Hello students, the point that will be continued in this lecture, that is the transcription. And in the previous lecture we have seen that is what is meant by transcription. Then the only one strand of the DNA is involved in the process of transcription. That strand we have given the name template strand, and the other strand which is not involved in the transcription but it acts as a reference strand, and that strand we have called as the coding strand, or we also call it as a sense strand. So these structures of the transcription includes different points, and the first point which we have also studied, that is the transcription unit. And we have seen the definition of the transcription unit is that the transcribed segment of DNA, the transcribed segment of DNA which forms the RNA is called as the transcription unit, which should have the three important components: promoter, structural gene and the terminator. The promoter is always present at the 5' end, the terminator is always present at the 3' end, and the structural genes are present in between this promoter and terminator, which are actually the genes which are responsible for the protein synthesis. Up to this we are clear from this transcription unit. But one important point that we'll discuss in this lecture from the same transcription unit, and that is the role of the enzyme in the transcription. So the point that will be continued in this lecture is the second point, and that is the transcription unit. From this point we have seen that what is promoter, what is structural gene, what is terminator, where the RNA polymerase bind. It is always bind to the promoter region which is present at the 5' end. We have also seen in this point that is a coding strand, non-coding strand, and now we are giving the emphasis or the focus on the enzyme. As we know, the enzyme which is involved in this transcription process, and the name of that enzyme is RNA polymerase enzyme. The name of that enzyme is RNA polymerase enzyme. But the major difference of this enzyme in prokaryotes and eukaryotes is that in prokaryotic only one RNA polymerase is there. In prokaryotes only one RNA polymerase is there for the synthesis of protein, or we say that for the transcription process. This RNA polymerase carry out the transcription in the prokaryotic by the using of only one enzyme, and the name of that enzyme is the RNA polymerase enzyme. But in eukaryotes, you will find three different types of this RNA polymerase which has role in the transcription, and these RNA polymerase are classified into RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. But here we have to say, or we have to see that which polymerase enzyme form the different types of the RNA, because the transcription means the formation of RNA, and as we know the RNA are of three types: mRNA, rRNA and tRNA. So all these three types of RNA are formed on the single strand of the DNA, and that process we have named it as a transcription. So in eukaryotes, three types of the RNA polymerase are involved, and these three types of RNA polymerase form different types of the RNA by the process of the transcription. So as we know, the prokaryotes has only one RNA polymerase that can form mRNA or RNA or tRNA from the single strand of DNA. But in eukaryotes, what we will find, this eukaryotes has three different types of RNA polymerase. This is RNA polymerase 1, then RNA polymerase 2, RNA polymerase 3. Okay, these are the three types of the RNA polymerase. Now we have to see which type of RNA polymerase form which different types of the RNA. So this RNA polymerase 1 forms rRNA. It forms rRNA. RNA polymerase 2 forms mRNA, which is also called as primary transcript, and hnRNA, that means heterogeneous nuclear RNA. In the next point I will discuss about this hnRNA, but here this RNA polymerase 2 is involved in the transcription or in the formation of mRNA and hnRNA. This mRNA and hnRNA are the two different things. Why? That we'll see in the processing of the mRNA. So here keep in mind that this RNA polymerase 2 is involved in the transcription of mRNA and hnRNA. And this third type, RNA polymerase 3, get involved in the synthesis of tRNA and snRNA. SN, the full form of this SN is small nuclear RNA. It is small nuclear RNA. Now I will explain this point, that is polymerase 1 form rRNA. As I told you that three types of RNA are there: rRNA, mRNA, tRNA. What is the function of rRNA? rRNA always attached to the ribosome and it is involved in the process of protein synthesis. mRNA, that is the messenger RNA, which is formed on the DNA strand and which carries message from the DNA to the ribosome for the protein synthesis. And tRNA are called as a transfer RNA, which are always involved in the process of transfer of activated amino acid to the ribosomes. And what is meant by snRNA? Because every student knows the three types of RNA, but number of students don't know what is meant by small nuclear RNA. Small nuclear RNA means these are the type of the RNA which are very small one, and generally these RNA are involved in the process of splicing. The same thing, that is the splicing, means the removal of intron. That we will see in the next part, that is the processing of the mRNA. So here only you keep in mind that is the small nuclear RNA is also a type of RNA which are generally involved in the splicing process, that is the removal of intron, which is generally carried out during the processing of the mRNA. Okay, so there is one trick. Then by this trick we can easily understand these three types of the RNA polymerase and which RNA they can be synthesized easily. So one trick is there, and that trick I will write here. All of you know the clone. Okay, all of you are well known about this clone term. Okay, what is clone? Clone, that term we have seen in our first chapter, reproduction in lower and higher plant. And in that chapter we have seen that the genetically identical individuals are called as the clone. Genetically identical individuals are called as a clone, which are products of the asexual reproduction, which are generally produced from the single parent. The clone, that means exact Xerox copy of their parents. Okay, the single individual from that clone, see here what I am saying, the single individual of that clone is called as a ramete. I am writing here what is a ramete. Ramete means the single individual of the clone. Clones means there are lot of or many identical individuals that we call as a clone, and from that many individuals, one individual which we taken outside, and that single individual is called as a ramete. So we have to understand these three type of RNA polymerase by using this trick. And how this trick can be useful? See here, R means rRNA. Here polymerase 1 is involved. M means mRNA. Here polymerase 2 is involved. T means tRNA. Here polymerase 3 is involved. Okay, this trick will be useful to you because the MCQ point of view, if question is asked for the NEET exam, that is the which type of RNA polymerase is involved in which type of the RNA formation, then you have to keep in mind the trick. The ramate first R, so RNA polymerase 1 for rRNA. Second is word M, so RNA polymerase second for mRNA. Third word is T for the RNA polymerase, third for the tRNA. So it is a very simple trick to memorize these different types of RNA polymerase which are involved in the transcription of these different types of RNA, and it will be found only in the eukaryotes, but in prokaryotic only one RNA polymerase is there which is involved in the transcription of the RNA. Okay. Now one more thing is that, what is that? That in these eukaryotes, the mRNA which are formed from the single strand of DNA that we have defined as a transcription, this mRNA is non-functional. Okay, what I am saying, the mRNA which are formed from the DNA. Okay, now I will write it differently so you can understand it easily. See here, this DNA strand forms mRNA. In center dogma we have seen this process is called as transcription. What is it called as a transcription? So I am discussing here that in eukaryotes, the mRNA which are formed from this DNA, is non-functional. It is not active one. Okay, so this mRNA should undergo processing or maturation. After processing on this mRNA, then and then only this mRNA become active one, and it will take part in the protein synthesis. This happen only in eukaryotes, but in prokaryotes, this mRNA which are formed from the single stranded DNA by the process of transcription is originally active one. It is originally functional one. So it do not undergo the processing like the eukaryotic cell, and that's why you will not find the mRNA processing in prokaryote, but you will find the mRNA processing in eukaryote. It is a major difference between these eukaryotes and prokaryotes. One major difference is that about the enzyme, only one RNA polymerase is involved in the prokaryotes, and the 3 type of RNA polymerase are involved in the eukaryotes. Second difference is that processing of the mRNA is not required in the prokaryotes, because mRNA which are formed from the DNA in generally bacteria or in other prokaryotes is functional. That's why it is immediately take part in the protein synthesis. But the mRNA which are formed in the eukaryotes from the single strand of DNA is a non-functional, so it should be undergo processing, and after the processing that mRNA become active one and it can take part in the protein synthesis. Then what is mean by processing? In detail this part we will see in the next part, but here keep in mind one thing is given in the textbook, that is in prokaryotic there are no intron. In prokaryotic there are no intron. That's why mRNA processing is not required in prokaryotics. The number of times the MCQ is asked that why in prokaryotic the mRNA not undergoes processing, but in eukaryotic it undergoes processing. The main answer to these points is that is intron. What is meant by intron? Intron means the non-coding sequence which are present on the DNA. The non-coding sequence which are present on the DNA get transferred on that mRNA, because mRNA form on the DNA. So we also say that the intron is the non-coding sequence on the mRNA. But this non-coding sequence of mRNA, this non-coding sequence that we call as the intron is present in the mRNA of eukaryotes, but it is absent in the mRNA of the prokaryotes. So in the process of processing, this intron codon from that mRNA should be removed. Here on this mRNA, introns are present, so they are removed in the processing process. But on this mRNA of prokaryotics, no intron is present. So there is no need of the removal of it, so no processing will be found here. Okay, so you have to also keep in mind why the processing is there in eukaryote and why it is not in the prokaryotic, because intron, which is a non-coding, non-essential part of mRNA, is not present in the mRNA of prokaryote, but it is present in the mRNA of the eukaryote. The same point we will discuss now, then you will get clear idea about the intron part of the mRNA. Okay, now the third point that now we are going to discuss is the transcription unit and the gene. Next point of the transcription is the transcription unit and gene. In this point, what we are clear that transcription unit require RNA polymerase enzyme. Question is asked for MCQ that which enzyme is involved in the process of transcription? RNA polymerase. But there are three types of RNA polymerase in eukaryotes, one type in the prokaryotes. Next, we have seen that prokaryotic do not have intron part, which is non-essential gene part of the mRNA. So it is not required the processing of mRNA, but here on this mRNA there will be intron part, so processing is essential. Now the third part, that is the transcription unit and gene, that now here we will discuss, and in this point we have to discuss what is meant by cistron, what is meant by gene, and what is meant by monocistronic and the polycistronic structure. So here what we see, okay, the next point is transcription unit and gene. Transcription unit and gene. Now we'll see first what is meant by gene. See here now the number of definitions in this point we are going to discuss. Gene means the sequence of DNA. See here, the sequence of DNA that codes for rRNA, mRNA and tRNA. Okay, that means suppose this is the DNA. Okay, this much is the sequence of DNA that we call as a gene. It is a long segment of DNA, but not all part of DNA are involved in the synthesis of mRNA, rRNA and tRNA. Only this much part of this whole DNA is involved in the formation of rRNA, mRNA and tRNA. So this much part of the DNA we call it as a gene. So what is meant by gene? Gene means the sequence of DNA that forms the code, means that information for formation of that particular RNA, the information which was present or which is present on this DNA, that is called as the code. So the segment of DNA that code for rRNA, mRNA and tRNA are called as the gene. So this definition is very important for one sentence of question, and you have to revise it. That is, the gene means a segment of DNA responsible for formation of rRNA, mRNA and tRNA. Then the second definition is this cistron. Second definition is the cistron. What is cistron? The segment of the DNA responsible for synthesis of the polypeptide chain. So cistron means segment of DNA. It is segment of DNA for synthesis of polypeptide chain. Here one word is important, that means polypeptide chain. That is polypeptide chain. Again, see here, if this is the diagram of the DNA, okay, not whole DNA is involved in the synthesis of polypeptide chain. There is some small fragment is there. Suppose this is a small fragment of this DNA. This small fragment of DNA is involved in the synthesis of polypeptide chain. So this small segment will be cistron. It will be cistron. So what is the definition of cistron? Segment of DNA for synthesis of polypeptide chain. And what is polypeptide chain? Polypeptide chain is nothing but the protein formation, because proteins are made up of the polypeptide chain. So the segment of DNA which is responsible for synthesis of protein, and that segment of DNA we say that is a cistron. So gene and cistron, these two important terms are here that we are discussing under the title Transcription Unit and the gene. Now we will see that monocistronic part of the mRNA and the polycistronic part of the mRNA, where these monocistronic genes are present and where the polycistronic genes are present. So next to this, that single structural gene in transcription unit. See here, single structural gene. That means monocistronic condition. Single structural gene, that is a monocistronic. Single structural gene is present in transcription unit of eukaryotes. Okay, and many structural genes, many means polycistronic condition is present in transcription unit, in transcription unit of prokaryotes. See here, now what is meant by the monocistronic and polycistronic condition? Here one thing is that the monocistronic means the single structure gene is present in transcription unit. Now we'll revise this term, transcription unit. Transcription unit means the segment of DNA which get transcribed. In other word, we call as a template DNA, or in simple meaning we call as a transcription unit, means the DNA which are going to form the mRNA. So on this DNA in eukaryotes, only one structural gene is present. On the DNA of the eukaryotes which are going to form the mRNA, on that DNA only one structural gene is present. One, that's why it is called as mono. Mono means one. Monocistronic condition. And many structural genes are present, that is called as many means polycistronic is present in the DNA of the prokaryotic. Just we have seen the definition of the cistron. Cistron parts means that part of DNA which form the polypeptide chain. So very simple meaning is there that on the eukaryotic DNA there is only one gene, and that gene we are calling it as structure gene. Then what is the function of this structural gene? This structural gene then going to form the single type of protein. Only one gene is there, and that one single gene or one structural gene going to form only one type of the protein, because single gene is there, one gene form one protein. Okay, so here the meaning mono means one and cistronic means the cistron part. That means one gene on that transcription unit or on that DNA form only one type of protein in the eukaryotes. But in the prokaryotes, there are already many structure genes are present in prokaryotes. So many structure genes form many different types of protein. So here we have given the term polycystonic. I think all of you get this point that monocystonic means only one structural gene in the eukaryotic responsible for formation of only one type of protein. So it is called as monocystonic. In prokaryotes, there are already many structure genes are present in prokaryotes which are going to form many types of the proteins. That's why it is called as polycystonic. So again, I will give you here a trick that for the MCQ point of view, students are always get confused that which gene is monocystonic and which is called as polycystonic, or the example of this mono and polycystonic we cannot get clearly. So we have to see the meaning of this, and that's why we have to follow one trick, and that trick is that simple trick is here. Prokaryotic stand for P and polycystonic also for P. So PP means prokaryotic polycystonic. Then ultimately you can recall that the monocystonic means eukaryote. So you have to keep in mind PP, polycystonic condition, is found in the prokaryotic. So it is very simple point that again here we differentiate this prokaryotic from the eukaryotic, that prokaryotic has polycystonic condition and eukaryotic has a monocystonic condition. Three differences we have already seen in between prokaryotes and the eukaryotes. One first difference is that prokaryotes have only RNA polymerase and the eukaryotic has the 3 RNA polymerase 1, 2 and 3. Prokaryotic has a do not intron, so no processing is there. Eukaryotic have the intron, so processing is there. Prokaryotic has the polycystonic condition and eukaryotic has the monocystonic condition. These 3 points are very important from the MCQ point of view. Now we have to see that in eukaryotes we have to see that there is the sequence of codon that we call as a exon. So I will write here the next point. Okay, this term is very important that I am going to write here. That is in eukaryotes there is interrupted non-coding sequence is present, which is called as introns, and the coding sequence or express sequence are called as exon. Only exons appear in mRNA of eukaryotes. These 3 points are also important. See here, in eukaryotes, now here we have not written the single word about the prokaryotes, all about the eukaryotes. In the eukaryotes, there are interrupted non-coding sequence, and that we call as introns. So this definition is very important. If they ask question that is what is intron, then you have to write the definition: interrupted non-coding sequence present on the DNA, present on the transcription unit, present on the template DNA is called as the intron. Okay. So what is meant by interrupted? Interrupted means on that mRNA or on that DNA in between, not continuous, in between. If it is intron, it is intron, it is intron. So in between the other segment, there are the segment of the introns are present. Interrupted means in between these introns are present, but what is the main function of this intron? They are non-coding. They do not form further any type of the protein. They are not involved in the synthesis of protein. So this non-coding sequence is very important. That is the sequence which is not useful for the protein synthesis, that we call as a non-coding sequence. So the interrupted, or if you not understand this word, you can also say that the non-coding sequence is called as an intron, and it is present on the mRNA strand, because from this mRNA the protein synthesis are formed. But if the non-coding, that means which do not have any role in the protein synthesis, and that sequence is present on the mRNA, then it is not useful. So that we have to remove from the eukaryotes. And just we have seen that it is not present already in prokaryotic, so it is not required to remove it from that prokaryotes. So it is a non-required part of the mRNA, and that we call as an intron, because it is non-coding. It is not useful for the synthesis of protein. And opposite to is coding. Coding means responsible for protein synthesis. It is non-coding, not responsible for protein synthesis. That is called as an intron. And then what is meant by exon? Opposite sequence, or in between the intron there is exon. And what is the exon? The coding sequence or expressed sequence is called as exon. The sequence of the mRNA which are actually form the protein synthesis, that is called as the exon. So opposite to non-coding, coding sequence. Opposite to intron, exon sequence. That means, suppose I will write here, suppose this is the mRNA. Okay, this is the mRNA. On this mRNA, this is exon. I will write simply EE. This is exon. And if we discuss it, it is an intron. So we have defined the definition of intron is, it is interrupted non-coding sequence. Interrupted means it is present in between this exon. So it is called as interrupted. It do not involve in the protein synthesis, so it is called as non-coding. But this exon part is called as a coding, because it actually going to form the proteins. So it is a coding sequence, that is the exon, and it is a non-coding sequence, that is the intron. These two terms are very, very important. That's why I am repeated it many times. That is what is exon and what is the intron. These two definitions are also important for theory point of view. And the last one is that only exons appear in mRNA, because we have to remove that intron. When we remove this intron from the mRNA, then and then only that mRNA becomes active one. Okay, and after removal of the intron, only exon part appear in the mRNA. That part is going to form the protein synthesis. That's why here we have said that exon appear eukaryotes. So this exon part is very important. And the last part of this point is the processing of hnRNA, heterogeneous nuclear RNA. That means on or from that mRNA we have to remove this non-coding part, that we have called it as a intron. So here we'll see how the processing of heterogeneous nuclear RNA takes place, and what is meant by heterogeneous nuclear RNA. So last part of this transcription is processing of hnRNA. hnRNA means heterogeneous nuclear RNA. Okay, but before this, or if we have to see what is meant by hnRNA. hnRNA is simply means the RNA which has both intron plus exon. That means it has both non-coding and the coding sequence. The such RNA which has both intron as well as exon, which is not processed. Before processing that, RNA is called as hnRNA, and after processing we call it as a mRNA. So it is very important what is meant by hnRNA. RNA which has intron as well as exon part. Okay. Another definition is there, that is primary transcript. primary transcript. Then what is mean by primary transcript? Form of RNA transcribed from DNA. Form of RNA transcribed from DNA, that is called as the primary transcript. That means the RNA which are formed from the DNA are called as primary transcript. So if the question asks for what is heterogeneous nuclear RNA, then we have to say that it is RNA which has both intron as well as exon, or in other words we said that RNA before processing is called as heterogeneous nuclear RNA, or in other words we say that heterogeneous nuclear RNA has not undergone the processing in eukaryotes. Okay. And what is meant by primary transcript? Primary transcript means the form of RNA which are transcribed or formed from the DNA, that is called as primary transcript. Now we'll see how the processing of this hnRNA takes place in the eukaryotic cell by the three processes, and those are the capping, then splicing and the tailing. So we'll see the processing of hnRNA by this diagram. Here in last lecture we have seen that this is our double-stranded DNA. One strand run as 5' to 3', and other strand runs as 3' to 5'. In last lecture we have seen that on the template strand which always run 3 to 5, there will be formation of the mRNA. So if it is RNA polymerase enzyme, from this mRNA is formed which has the polarity that is 5' to 3', because in the replication point also we have seen that new DNA or the new RNA is always formed in the direction 5' to 3'. Okay, this 5 is called as a head and this 3 is called as a tail. Head is the 5' point and tail is the 3' end. Okay, so this RNA or mRNA always form 5' to 3' on this template strand. Okay, so on this mRNA, this is called as the mRNA. How we will find the sequence? This is called as intron and this is called as the exon. Okay, this mRNA which has this intron part and this is exon. We have to remove this intron part. So first process in this processing is that at the head we always add the cap. At the head we always put the cap. So first point is that at the 5' end of this newly formed RNA there will be the capping, or capping means the addition of methyl guanosine triphosphate. See here, methyl guanosine triphosphate. The methyl guanosine triphosphate get added at the 5' end of the mRNA, and that process is called as the capping. That process is called as the capping. Methyl guanosine triphosphate get attached at the 5' end of the newly formed mRNA. It is called as the capping. Then next step is that 5' to 3'. We have to remove this intron because these are the non-coding sequence. Okay, it do not form the any protein, so this non-coding sequence should be removed. And removal of this intron. See here, these are the introns. We have to remove this intron. This is methyl guanosine triphosphate. Okay, and we have to remove this intron. So removal of intron is called as splicing. Removal of intron is called as splicing. because here we have removed this intron part in the second step. And the third step is tailing. The third step is called as tailing. What happens in tailing? In this tailing process, here this 5' end, here this 3' end, methyl guanosine triphosphate are added here, and at the 3' end there is the addition of poly A. Poly A means adenine base pair, but it is many, so it is called as poly A tail are added at the 3' end, and that process is called as the tailing. Many adenosine bases are added at the 3' end, and third process is called as a tailing. So the mRNA which are formed, so this mRNA which is formed from this DNA should undergo three processes, that is the capping, splicing and the tailing. And after capping, splicing and tailing, we may say that this mRNA is functional one which can easily take part in the protein synthesis, or which can easily carry out the process of translation. But what is meant by capping, what is meant by splicing, what is meant by tailing, these definitions are also important. If the question is asked what is meant by capping, capping means, the definition is that at the 5' end of the mRNA, at the 5' end of the mRNA, there will be addition of methyl guanosine triphosphate. It is called as the capping. Okay. Second point, what is splicing? Splicing means the removal of the intron. The removal of the intron from the mRNA is called as the splicing. And what is meant by tailing? Tailing means the addition of polyadenylation or addition of poly A, many adenosine at the 3' end of the mRNA is called as the tailing. So these definitions are very important: capping, splicing and the tailing. This is the sequence. First there will be capping, then there will be splicing, and then there will be tailing. Okay, capping always takes place at the 5' end of the mRNA. Tailing always takes place at the 3' end of the mRNA. And simply splice means removal of the intron is called as a splicing. So this is very important part of the processing of the hnRNA or the processing of the mRNA in the eukaryote, because we have seen that this intron part is not present in the prokaryotes, so processing is always required in the eukaryotes. And here we have seen that this much part of the processing of the hnRNA. I will repeat some points. That heterogeneous nuclear RNA means the RNA or the mRNA before the processing is called as heterogeneous nuclear RNA. When the capping, splicing and tailing takes place in this hnRNA, then that process is called as the processing or the maturation of the RNA. Whole this process of the maturation takes place in the nucleus itself, because nucleus is that region where the DNA forms the mRNA by the process of transcription. So whole this process is carried out in the nucleus. And as I told you that 5' to 3' is the polarity where there is the formation of the always new DNA, always new RNA. So this RNA which is always formed in the direction 5' to 3'. If 5' end is considered as a head and 3' is considered as a tail, then cap is always added at the 5' end. So it is the capping at the 5' end, and tail is the 3' end. So polyadenylation or the tail get added at this 3' end. So this is about the processing of the hnRNA. Now students, in this lecture, what we have seen today? We have seen the role of 3 RNA polymerase in the eukaryotes and role of only 1 RNA polymerase in the prokaryotic. Ramid is the trick to identify these 3 types of RNA polymerase. 1 polymerase for rRNA, 2 polymerase for mRNA and 3 polymerase for the tRNA. Then we have seen that the processing is required only in the eukaryotes because it has intron, and the processing is not required in prokaryotes because it do not have the intron. Then we have seen the cistron and the gene definition. Cistron means the part of the DNA responsible for the polypeptide synthesis or the protein synthesis, and gene is a part of the DNA which is involved in the formation of rRNA, mRNA and the tRNA. Then we have seen how the processing of this hnRNA takes place by the 3 methods: capping, splicing and the tailing. Capping always occurs at the 5' end by addition of methyl guanosine triphosphate. Splicing means the removal of the intron from that exon part or from near the exon part from the mRNA, and tailing means this addition of the poly A many adenosine bases to this 3' end. Thus here we have completed the transcription point. Questions are always asked for the long question for 4 marks, that is what is meant by transcription, describe the process in detail. Then you have to complete that point by 4-point transcription, then transcription unit, and the gene, and the last part is the processing of the hnRNA. After that we will discuss the genetic code, and after discussing the genetic code we will clear how the translation occurs in the eukaryotes. Okay, I think all of you get this transcription process very clearly. It is also important for MCQ point of view as well as for theory point of view. Okay students.